Alright, now ALT, alanine aminotransferase or alanine transaminase. It is a commonly raised investigation in patients to check for liver disease. It is also known as alanine transaminase or serum glutamate pyruvate transferase. The word transaminase means an enzyme that transfers amino group from one molecule to another. It is found in skeletal muscles and in the liver in higher concentrations. ALT is found in cytosol of hepatocytes, that is inside the liver cell. So if it leaks out into the plasma, it symbolizes liver cell damage. Here's the structure. It is a polypeptide enzyme that cuts amine group from one amino acid and transfers it into another amino acid. It converts glutamate to alpha ketoglutarate in muscle, enhancing glycolysis and production of energy. It converts alpha ketoglutarate to glutamate to improve gluconeogenesis in the liver. Alanine is the molecule that gets transferred from muscle to liver through blood. Thus, the enzyme increases the blood glucose level. There are hardly any bacterial infections of the liver because of its high bile content. While bacteria thrive on high glucose levels, there is no such establishment on viral growth and glucose. So we can only imply that virus infected cells are destroyed and ALT comes out into the plasma as cytoplasmic content. Alanine aminotransferase levels increase in order to combat intramuscular hypoglycemia. Clinically, increased alanine aminotransferase signifies hepatocyte injury. In many diabetics, ALT levels increase as a result of protein breakdown in hypoglycemia of the muscle. Diabetes is a skeletal muscle disease in which the glucose does not enter muscles due to a defect in insulin. This is due to a desensitization of insulin receptors. Moderate levels of exercise should remove this problem as it decreases the basic glucose level in the muscle and resensitizes the insulin receptors. Hypoglycemia and diabetes in the muscles due to desensitization of insulin receptors causes proteolysis, which can potentially release ammonia. Ammonia is toxic, so the enzyme transaminates it in the muscle and sends it to the liver as alanine through the blood. Moderate increase in ALT may signal towards proteolysis as an indication of muscular hypoglycemia. Exercise could be recommended for the resensitization of insulin receptors. I was thinking of stimulating ALT. This will cause hypoglycemia in the blood and is of no use. If we stop ALT in the muscle, then there is a chance of stopping proteolysis due to a local negative feedback mechanism, but we will have an increase in ammonia, which is toxic. So ALT will be a very indirect method of controlling diabetes, and use of ALT as a drug mechanism is far-fetched. We may use ALT in different tissues to remove ammonia content. Ammonia can be removed from various tissues in conditions like liver disease, which will affect the brain through ammonia toxicity, and especially in Helicobacter pylori infection as an oral drug. It may also imply that ALT that is released into blood in hepatocyte injury could be to combat the ammonia that is caused during inflammation. Ammonia thus excreted in the blood to the liver undergoes urea cycle to convert into urea, which is excreted in the urine. Thank you. Like, share, subscribe and hit the bell icon.